आज ही डाउटनेट ऐप डाउनलोड करें। डाउटनेट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सोल्यूशन पाओ। डाउनलोड नाउ।
Hi, in this question we are asked to explain replication was allowed to take place in presence of radioactive denucleotides. So here what they have done, replication has been allowed. Okay, I'll first read out the question. Replication was allowed to take place in presence of the radioactive deoxynucleotide, okay, precursors in E. coli that was mutant for the DNA ligase. So newly synthesized radioactive DNA was purified and the strands were separated by the process called as the denaturation. There were centrifuged using density gradient centrifugation. Which of the following results would be correct? Okay, so let us understand the questions first. So here they have, what they have taken? They have taken the radioactive deoxynucleotide in the E. coli and made it to replicate, and these newly synthesized DNA, they were purified with the help of the denaturation process. Then the whatever the DNA it is formed, it has been centrifuged using a density gradient centrifugation. So we have to identify which of these graphs are correct related to this question. So in order to understand that, first let us consider the process of DNA replication here. So here in the first process, the DNA is, let us consider DNA molecule here, and this DNA molecule contain one of the strands as a radioactive, that is N14 molecules are present is DNA strand. So what happens during replication here? So both the strands will be separated, carrying one of the strands carrying N4, other one is carrying the heavy DNA molecule. Okay, so the newly synthesized molecule of the strand will contain the, here the older or the parental strand I am writing in red and the daughter DNA I am writing in green. So this newly synthesized strand will contain the 50% of the radioactive DNA, 50% of non-radioactive DNA. This is in case of the first generation, or we carry it as a generation 1. If it is continued further in the generation 2, which results in the formation of the 4 daughter strands or the daughter DNA molecules here. So I am representing here the 4 different daughter strands 1, okay, and here, okay. So here then there is a formation of the daughter strands here, and then I am taking the another daughter cell and allow it to synthesize itself. It forms a daughter DNA, and then one more strand of the DNA, it synthesizes one more daughter DNA, and here one of the radioactive strand, another one is the non-radioactive. So here once you observe here, there is a formation of the 2 or the 50% of the radioactive DNA and 50% of the non-radioactive DNA, because it is a semi-conservative mode of DNA replication. We can, where the parental DNA is conserved for the further process. So here you can, as you can observe in the generation 2, this 50% of the radioactive DNAs, whereas the 50% are the non-radioactive DNA. So by observing this, you can finally come to the conclusion that the option A is the correct answer, because here you can see the 2 peaks. Okay, so the 2 peaks are present where 50% of the molecules are. So therefore the option A is the correct answer.
क्लास 6 से 12 से लेके नीट, IIT, जेई मेंस और एडवांस के लेवल तक 10 मिलियन से ज्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनेट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।